Hi folks, welcome to AP Biopharma with Dr. D. This is video number 2 in this lecture series of how structure determines function, a tale made of biomolecules. And in this video, we're going to talk about functional groups. What are they and why do we need to recognize them? So carbon compounds are really interesting because they contain functional groups. So what are functional groups? Functional groups are groups of 2 or more atoms that have specific chemical properties. They're really parts of an organic molecule which are involved in chemical reactions. They give the organic molecules their distinctive properties. And the major functional groups we're going to be talking about are hydroxyl, carbonyl, carboxyl, amino, sulfhydryl, phosphate, and methyl. They affect the reactivity of a molecule. For instance, if you take a hydrocarbon, which is hydrophobic, and add a few hydroxyl groups to it, now this hydrocarbon becomes hydrophilic. So you can increase the solubility of a molecule in water by simply adding a hydroxyl group. So let's look again at the differences that we circled in last video. Let's look at them again. So first there is this difference between glucose and galactose. Then we have these two are identical between glucose and galactose, but there's something different in fructose, right? So these circles really are functional groups. Their presence or absence and their positions will really determine the properties of the molecule. So let's look at another example of a difference between functional groups. Identical carbon skeleton, CH, 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 CH. The only difference between estrogen, which here is shown as estradiol, and testosterone. Testosterone is the male sex hormone, estrogen is the female sex hormone. The only difference is the functional groups that are circled either in red or blue. In one case, it's a hydroxyl group, in another case, it's a ketone group. That single difference makes all the difference, as a female versus a male lion can tell you. The major functional groups are the negatively charged group carboxyl and phosphate, the positively charged amino group, and the neutral but polar groups hydroxyl, sulfhydryl, carbonyl, and aldehyde. And you're going to learn to recognize them. Let's talk first about the non-charged but polar groups. The first one is the hydroxyl group. It is oxygen linked to a hydrogen. It can be written OH or HO. The presence of hydroxyl group in a hydrocarbon turns that hydrocarbon into an alcohol. The name ends in OL. Ethanol is the most famous of all alcohols because it is present in alcoholic beverages. The next group we're going to look at is the sulfhydryl group. It has a sulfur and a hydrogen attached to it. Sulfhydryl groups are present in 2 amino acids and are very important in maintaining protein structure, as we're going to explore later. And finally, we have the carbonyl group, which is a carbon linked via 2 covalent bonds to oxygen. So it's C double bond O. Now the carbonyl group comes in 2 different flavors. If it is in the middle of the molecule, it's called a ketone. In this case, what is shown here is acetone. Acetone is a major component of nail polish remover. On the right, it's propanol, which is an aldehyde, because the same functional group, a carbon with a double bond O, is at the end of the molecule. So if it is at the end, it's an aldehyde. If it's in the middle, it's a ketone. Now we're going to talk about the molecules that are considered acids. Molecules that are considered acids have acidic functional groups. The functional groups are negatively charged. And if a molecule has a negatively charged functional group, such as carboxyl or a phosphate group, that molecule is considered an acid, and it's in its name, nucleic acid. It has phosphate functional groups. Fatty acids have carboxyl functional groups. Amino acids have carboxyl functional groups. Why are they called acid? Because the functional group ionizes in solution and it loses a hydrogen ion. Therefore, it increases the hydrogen ion concentration and lowers the pH. And let's look at the carboxyl group. The carboxyl group is a carbon, double bond O, and then there's a hydroxyl group right here. Now you need to be careful, but these are not two separate functional groups. This is not an aldehyde and hydroxyl group. This is a carboxyl group. So the carbon is linked to two oxygens. Normally, this is what it looks like. However, once a compound containing this carboxyl group is dissolved in a water-based solution, it ionizes and it becomes O negative here. So it's negatively charged, and it loses a hydrogen ion, positively charged. So it increases the hydrogen ion concentration, thereby lowering the pH of the solution. Similarly, a phosphate group actually loses two hydrogen ions and has two negative charges. Is negatively charged in solution because it has donated its hydrogen ions. Remember, these groups are negatively charged, and if they're negatively charged, they are acidic because they have lost hydrogen ions. They have increased the hydrogen ion concentration in the solution. Now, we also have a positively charged functional group, the amine group, and the amine group is basic. So amino acids are really both an acid because they have an acidic group, and they're a base because they have an amino group. An amino group really is a nitrogen linked to two hydrogens. However, in solution, a compound that has an amino group usually picks up a hydrogen ion from the solution, and now you have NH3. Three hydrogens are bound to nitrogen, and now there is a positive charge on the nitrogen. This is the ionized form of NH2. It's sometimes written as NH3 plus, and this acts as a base because it removes hydrogen ions from solution. It lowers hydrogen ion concentration, thereby increasing the pH. And here's another example. It can be either NH3 plus or NH plus right here. These are two examples of ionized amino groups in a molecule. They're also nonpolar and non-charged functional groups, and they're considered hydrophobic. The most simple functional group is the methyl group. The methyl group is something that comes from methane. Methane is CH4. The functional group is just a carbon with three hydrogens, CH3. CH3 can be added to a number of compounds, and if you have methyl groups, they're not charged, and they confer hydrophobicity. Molecules with methyl groups are hydrophobic. Okay. Amino acids are the monomers of proteins. They contain both an amino group and a carboxyl group. There's a negative charge on the carboxyl group and a positive charge on the amino group. Amino acids have a central asymmetric carbon to which we have an amino, a carboxyl group, a hydrogen atom, and an R group. This R group is different for every amino acid, and it is responsible for its unique properties. The R group gives the chemical properties to each different amino acids. The R groups themselves can be grouped in several different categories. They can be nonpolar, they can be polar uncharged at physiological pH. Physiological pH is pH around 7. They can be polar negatively charged, that means they're acidic, or they can be polar and positively charged, that means they're basic. And that depends on what kind of functional groups they have. Here are some nonpolar hydrophobic amino acids. Why are they nonpolar and hydrophobic? Because their side chains, their R groups, are nonpolar and hydrophobic. And the side chains, the R groups, are here highlighted in white. For this particular one, the side chain is a simple hydrogen. This is a methyl group. This is a combination of methyl groups, CH, CH, CH. All you see is CH. These amino acids, their side chains, are nonpolar and hydrophobic. The polar amino acid contain side chains which can have some kind of polar functional group. Usually it's a hydroxyl group. Here in serine, you will see a hydroxyl group, a thiol group, sulfhydryl group, hydroxyl group, an amino group, and so on. You also have acidic, negatively charged ones, and basic, positively charged one. These two contain a carboxyl group. These contain amino groups. So the functional groups within the R groups, if they are polar, then the amino acid is polar. If they're acidic, the amino acid is acidic and negatively charged. If they're basic, the amino acid is considered basic and positively charged. Is this organic chemistry? This is the end of our lecture on functional groups. We will explore more functional groups and especially R groups of amino acid when we talk about the structure of proteins. This is for now. See you next time.